Hi everyone, welcome to the session. This is the seventh session in the MD 6028 Virology module. So we're kind of well into the second portion of the module now, in which we're looking specifically at the ability of viruses to cause disease. So we're looking at viruses as agents of disease. Another way of wording that is by saying we're looking at viruses as human pathogens. The fundamental principle is the same. We're looking at how viruses cause disease in humans. In the first portion of the module, we really spent quite a bit of time establishing what viruses are, defining viruses, looking at in quite a bit of depth at how viruses replicate, for example, and how the different groups of viruses replicate and how we separate viruses into groups. And hopefully that was really informative and really useful. And what we're doing now is we're applying this knowledge. We're using that knowledge to inform our understanding specifically of how viruses cause disease. And again, that's what we're going to be doing today. The aspect of this we want to look at today is a group of viruses called bacteriophages. So bacteriophages, in some people's heads, they can seem like a distinct type of microorganism. So you can have viruses and bacteriophages. However, the first thing we want to establish is that bacteriophages are viruses. They're just a subgroup of viruses. So what we want to do now is look specifically at this subgroup and how this subgroup causes disease. The reason we want to do this is because bacteriophages cause disease in a very specific way and in a way that is quite different from most other viruses. Now, we've talked quite a bit about how other viruses cause disease, and we'll talk about that more in the upcoming lectures. Most of you will have talked about it in the medical microbiology module as well, MD 6030. So what I want to do now is just tackle this quite niche portion of virology that's a little bit separate from the other viruses. As you'll see, the general principles still apply, but you'll also see that there are some unique things going on with bacteriophages that we really want to understand, because even though this is a bit of a niche area, we're talking about some quite serious diseases that are caused by bacteriophages, so it is important we understand this. So the first thing we want to do is establish what bacteriophages are, make sure we have an actual understanding of bacteriophages. And again, the first thing to emphasize is that bacteriophages are viruses. They're a group of viruses. They fit the criteria to be defined as viruses. And again, in the earlier lectures in the module, we spent quite a bit of time defining viruses and exploring what a virus actually is. And by all those definitions, bacteriophages can be classed as viruses. We even classify them in the same way. So we can classify them using Baltimore system, and we can classify them using phenotypic systems as well. However, they are slightly separate. Or I shouldn't say they're separate from viruses. They are viruses, but they group together within viruses. So they're a specific group of viruses. And why is this the case? Why do we group bacteriophages together? Bacteriophages essentially have a common ability to infect archaea and/or bacteria. So unlike most viruses, bacteriophages will infect members of the archaea and bacteria domains of life. So if you remember the domains of life, I'm sure you will have learned at some point, and we have three domains of life. We have eukarya, we have bacteria, and we have archaea. Now, multicellular life, eukaryotic life, belongs to the eukarya domain. Unicellular life, prokaryotic life, belongs to the archaea and the bacteria domain, and these are the domains that bacteriophages infect. So this is why we group them together. We also group them together based upon a very distinct an unusual structure they have. So we talked about how viruses have, so the general structure of a virus is a genome wrapped in protein. Okay, sometimes there's an envelope as well, but fundamentally a genome wrapped in protein. Bacteriophages fit that criteria, so they do fit. They do have a genome and it is wrapped in protein. However, as you'll see, the overall structure of bacteriophage has some other elements that are quite unique, that are quite distinct, and that mean it makes sense. They mean it makes sense to classify bacteriophages in a group because they have this really, really unique structure. So their unique structure and their ability to infect archaea and bacteria means we group these bacteriophages together, and that group is a kind of subgroup of viruses because they are viruses at the end of the day. As they are viruses, they are also obligate parasites. So we said all viruses are obligate parasites, therefore bacteriophages as viruses must be obligate parasites, and that's true. So the only way these bacteriophages can replicate is to infect a host. It's just for these bacteriophages, the host is an archaea cell or a bacteria cell. So they are using prokaryotic cells as their host rather than eukaryotic cells. On the right here, we have a diagram showing a bacteriophage, and it should be pretty clear that the structure here is quite different from the structure of the other viruses we've looked at so far in the module. Notably, we have this tail and these protrusions that look almost like arms and legs. Quite distinct, right? It doesn't look like the viruses we've looked at so far. The capsid, however, the very top of the bacteriophage, the head, this will look a bit more familiar because this is not dissimilar to the capsid of the other viruses. It's just the protein shell, and this is the portion of the bacteriophage within which the genome is stored. So again, that should look pretty familiar, but the rest, it might look a bit bizarre, and it is a little bit bizarre really. Again, it's essentially it's referred to as a tail, and there's this little collar connecting the capsid to the head, and this tail has these strange protrusions. Why is the bacteriophage shaped like this? The reason the bacteriophage is shaped like this is because it has a slightly different replication cycle to other viruses. We talked at length how viruses enter into the host cell, and that's absolutely true. However, for bacteriophages, the situation is slightly different because it's just the genome that enters into the host cell. So, and you can almost imagine this happening when you look at the structure. What happens is the bacteriophage lands on the outside surface of the host cell. So it attaches to the outside surface, nothing too unusual there. And then the genome is essentially injected into the host cell. So it's injected from the capsid through the tail into the host cell, and then replication can occur within the host cell. So it's quite different to the other viruses, but the end point is kind of the same because the end point is the genome being released into the cell. Now, for most viruses, that involves the virus entering into the cell itself and then breaking apart, allowing the genome to be expressed and replicated. But again, for the bacteriophages, it just involves an injection of the genome into the cell. Why have bacteriophages evolved to infect this way? You know, it's hard to say for sure. All we can really say is that prokaryotic cells are fundamentally different to eukaryotic cells. So bacteriophages have evolved to infect these prokaryotic cells, which are fundamentally different to the eukaryotic cells that the other viruses have evolved to infect. So it's perhaps not that surprising that differences in the mechanism of infection have arisen. Again, because the cell type being infected is fundamentally different. Prokaryotic cells and eukaryotic cells are really quite different to each other. This is a distinction I'm very sure you're aware of. If you're not, I would encourage you to spend a little bit of time going back over your notes from past years and really understanding the difference between prokaryotic and eukaryotic cells. So bacteriophages, as we've said, they're viruses, so they can be classed based on their phenotype, or they can be classed by the Baltimore classification system, really. They do share phylogenetic relatedness. However, like other viruses, they can't be mapped onto the tree of life. You can't class them higher than order. And again, this is something we've talked about for the other viruses, and it applies to the bacteriophages too. What can be done is a lot of bacteriophages can be grouped into families. So we can take bacteriophages and we can group them into kind of families, which are groups of relatedness. So these are phylogenetic families based upon phylogenetic similarity. However, we're generally not sure how these families relate to each other. And you can see this term on the slide here, Incerta sedis. And when you're looking at the classification of these bacteriophages and a lot of other viruses, actually you'll see Incerta sedis at some point in the classification, and it basically means unknown. We don't know this classification, so the classification hasn't been placed. And some of these families you can be classed into orders, but most can't really be classed much further than families. And even the ones that can be classed into orders, there's no real understanding of, again, how there's no understanding beyond order, and there's no understanding of how the orders and the other families relate to each other. It's just quite confusing, really. And again, we went into this at length when we were looking at the other viruses, and the principle here is the same. Okay, Carda virals, this is order here. As you can see, we have 3 families here listed within the order, and they all have this distinctive structure we talked about. You can see there's variations on this structure, right? There's variations in how long the tail is, there's variations in the appendages, so in these protrusions, but fundamentally they have this fairly common structure. They have this capsid at the top, then they have this tail, so this long tail that is used typically to inject the bacteriophage DNA into the host cell. And here we have an example of what I was just saying, really. So we have Podoviridae, which is a family of bacteriophage, and we have its phylogenetic classification. It's phylogenetically classified into this order Cordovirales. Above that, it's Incerta sedis. It's placement unknown. It's unknown classification. I think literally that's what it means, placement unknown, in Latin, or something very similar to that. It essentially means we don't know how to classify it. So that's what, if you're seeing this term Incerta sedis, that's what the fundamental meaning is. And again, you'll ultimately see this if you go higher than order for any bacteriophage, because they are viruses that we can't classify. In terms of the terminology, when we're describing the bacteriophage itself, we're using similar terminology to the other viruses because they are viruses. So the viridae is the suffix meaning family, and virals is the suffix meaning order. The term bacteriophage itself essentially means bacteriophage, bacteria eater. Bacterio from bacteria, phage from to eat, which is the term you're probably familiar with. Macrophage is a big eater. It's a large cell that eats, that also eats large elements or cells. That's what phagocytosis is, right? It engages in the process of phagocytosis. So yeah, that's why they're called bacteriophages, really. Okay, so that's a bit of an introduction to what bacteriophages are. Hopefully everything there is clear. Again, we're just talking about a subgroup of viruses that have some distinct characteristics, and because of those distinct characteristics, we group these bacteriophages together. What we want to do now is look at some of the different types of bacteriophages, and namely we want to look at virulent versus temperate bacteriophages, and I'm going to explain in the next section what those terms mean. Fundamentally, these are terms that are important to how bacteriophages cause disease. So as we're talking through these different bacteriophage life cycles, because that is what these terms mean, we have a virulent life cycle and a temperate life cycle. We're talking through this information because it's relevant to how these bacteriophages cause disease, which is something we'll be getting into when we get to the middle of the lecture. The two life cycles I'm referring to are the lytic and the temperate life cycles, also sometimes referred to as the virulent and the temperate life cycles. So we can roughly split bacteriophages up into those that engage in lytic life cycles and those that engage in temperate life cycles, but those that engage in temperate life cycles can often also engage in lytic life cycles as well. The principle of the lytic life cycle, also being referred to as the virulent life cycle, is that if a bacteriophage is infecting a prokaryote and it is causing lysis in that prokaryote, and again, that's what the lytic life cycle means. It's lytic, it's causing lysis, it's destroying the cell. We can justifiably define that as being a virulent life cycle because the bacteriophage is causing, fundamentally we can say it's causing disease. What it's really doing is completely destroying the cell, but it's analogous to disease, right? It's causing damage to the cell, it's causing pathology to the point that the cell is lysed. So the lytic life cycle is necessarily virulent because it destroys the cell that is being infected. We also have non-lytic life cycles, however, and again, a lot of bacteriophages will be able to engage in lytic or temperate life cycles, so they'll be able to do either. Temperate life cycles are life cycles that don't cause lysis of the host cell, so they don't destroy the host cell. We can refer to this, as opposed to the lytic life cycle, we can refer to it as the lysogenic life cycle. And again, we classify the bacteriophages that engage in this life cycle as being temperate. This is the really important life cycle when it comes to human disease. The lytic life cycle, so the virulent life cycle, is the life cycle that's destroying the prokaryote, so it's destroying the cell that's being infected. However, the temperate life cycle, the lysogenic life cycle, doesn't destroy the cell, as we said. However, what does happen is the bacteriophage genetic material is retained inside the cell. So infections occurred, the bacteriophage has injected its genome into the cell. However, the cell hasn't been lysed, it's still replicating, it's still alive. Okay, the issue there in terms of human disease is that very often the bacteriophage will introduce into the cell, as part of its own genome, specific genes that cause the prokaryote that is being infected to become virulent towards humans. So we have, and we're talking about bacteria here, because even though bacteriophages can infect archaea, when we're talking about human disease, it's bacteria that cause these human diseases that we're talking about here. So we have a normal bacteria, a healthy bacteria. It becomes infected with a bacteriophage. This bacteriophage injects its genome into the bacteria. Within that bacteriophage genome, there can potentially be specific genes that, when expressed by the bacteria, cause that bacteria to become pathogenic to humans. So prior to the bacteriophage infection, that bacteria wasn't pathogenic to humans, but now it's acquired these pathogenic genes from the bacteriophage. The bacteria is capable of causing human disease. This process of acquiring these genes from the bacteriophage is called lysogenesis or lysogeny. What's essentially happening is additional genes are being added to the bacteriophage genome. On the right here, we have a diagram of the life cycles of a temperate bacteriophage. So this is a bacteriophage that can engage in both of the life cycles we've discussed. So it can engage in a lytic cycle, and it can also engage in a lysogenic cycle. So first we'll look at the lytic cycle, and you can see the bacteriophage is attaching to the outside of the prokaryotic cell, and then it's injecting its DNA into the cell. Once the DNA is inside the host cell, it is then expressed, it is replicated, and these are the necessary steps to produce new bacteriophages. Again, this is analogous to the replication cycles of the other viruses that we've looked at in past weeks. We're looking at a very similar process because these are viruses after all, remember. So these new bacteriophages are being produced. You can see the term phage on the slide. The term phage is sometimes used in place of the term bacteriophage. It means exactly the same thing. It's essentially shorthand. So rather than saying bacteriophage over and over again, sometimes the word phage is used. You can see lysis then occurs when these new bacteriophages have been produced, and this lysis it releases these bacteriophages into the extracellular environment. These bacteriophages can then infect new host cells. So again, we're talking about single-celled microorganisms, so they're not going to be cells within the same host. They're going to be new host cells that are in the local environment. And again, this is why we call it the lytic cycle, because it's lysing the cell. On the other hand, we have the lysogenic cycle. So this bacteriophage and other temperate bacteriophages can also engage in the lysogenic cycle instead of the lytic cycle. The initial stage is the same. We have the bacteriophage attaching to the outside of the prokaryotic cell and injecting its genetic material into the host cell. This is where the cycles differ. So we can see on the right hand side, we then have a process called integration. It's not the same process as what we looked at for retroviruses. It's not that there's no retroviral integrase, there's no reverse transcription, any of those linked processes. We're looking at something that is, in terms of the process, very, very different. It's just named the same thing because the end point is the same. The integration is the same. If you remember the retroviruses, the end point of integration is a DNA copy of the viral genome being integrated into the host cell genome. Here, the end point is the DNA bacteriophage genome being integrated into the host cell genome. So the end point is the same. That's the takeaway here. It ends with the viral genome integrated into the host cell genome. When we were talking about retroviruses, you remember we then termed that integrated retroviral genome in the host genome as being a provirus. So we use the term provirus. The term we're going to use here is prophage, and prophage is just a specific term we use when we're talking about this integration, when it's being carried out by bacteriophages in a host cell. Essentially, then everything can go on pretty much as normal. The bacteria can replicate, it can continue to thrive, it's not killed, it's not lysed necessarily. There can be a potential switch down the line to lytic cycle, that's absolutely fine, but generally in this lysogenic cycle, the cell is continuing to live, it's continuing to thrive, it's continuing to divide, and importantly, it is retaining the bacteriophage genome within its own genome. So it has acquired new genes from the bacteriophage. And as we're going to look at in a bit, some of those genes can be genes that are responsible for pathogenesis. So by carrying those genes, the bacteria can gain the ability to cause disease in a host. Just a bit more information here on the lytic cycle specifically. So the lytic cycle again ends up with the infected cell being lysed. There's no integration. So when we were just talking about the lysogenic cycle, we were saying the bacteriophage genome becomes integrated into the host cell genome. That's not true in the lytic cycle. So that's specifically the lysogenic cycle. In the lytic cycle, there's no integration. Therefore, there's no prophage. There's no prophage formed because the prophage is the bacteriophage genome integrated into the host cell genome. Lysis is induced, as we said, and the consequence of this lysis is that huge numbers of bacteriophage particles are released into the local environment and they can then potentially infect nearby cells. So again, it's just a strategy for the bacteriophage to spread. This idea of viruses causing cell lysis to promote their own spread is not a new concept. It's something we've discussed in the earlier lectures. We looked at a lot of viruses that infect host cells that cause lysis of the host cells, and by causing lysis they release a large number of infectious virus particles into the environment. Same thing is happening here. Again, it's something we've touched on before. It's just here we're looking at bacteriophages doing it. How do these bacteriophages induce this lysis? So there's two proteins, there's lysines and there's holins. These are just proteins that are encoded by the bacteriophages that directly induce cell lysis. We don't need to go into too much detail about the mechanisms of that, although it would be useful to know these groups of proteins because these are the proteins that are directly responsible for inducing lysis. Now the temperate bacteriophages. So as we said, temperate bacteriophages can conduct either of these processes usually. So they can engage in a lytic life cycle and they can also engage in a lysogenic life cycle. This is quite normal for these temperate bacteriophages. The lysogenic life cycle is the life cycle to which the term prophage applies because we see integration of the bacteriophage genome into the host cell genome. Now there's a consequence of this, and as we said, the consequence of this is that specific genes can be introduced into the host cell genome. So the cell that is being infected can have its genome modified. It can have additional genes introduced. This is a type of horizontal gene transfer. So we can think of the different types of gene transfer that can happen. If we're thinking about reproduction and offspring being produced that are genetically non-identical to the parents, that's vertical gene transfer, right? Genes are being passed vertically down generations. This is an example of horizontal transfer because it's not going down generations, it's going across members of different species but not necessarily of different generations. So we think of it as being horizontal rather than vertical. There's other examples of horizontal gene transfer that I think we might not have talked about in this module, but anyway, there are other examples of horizontal gene transfer that exist. The main example of vertical gene transfer is just reproduction. Okay, again, so this occurs naturally. Yeah, this is a natural process. I've mentioned that temperate bacteriophages can be more interesting from a research perspective. And we're going to look at this in the last couple of lectures in the module actually, because the ability of temperate bacteriophages to introduce new genetic material into a cell and to do so in a stable way, so to introduce those genes into the host cell genome and to integrate those genes into the host cell genome is very useful from a research perspective. So by harnessing bacteriophages, and we do this with retroviruses as well, because again, retroviruses are capable of integrating their genome into the host cell genome. And for both these groups, we utilize this ability to conduct specific experiments that we otherwise wouldn't be able to do. So it's actually quite difficult to, within a lab environment, just introduce new material into cells and have that material integrated into the host cell genome. There's not really there's ways to introduce genetic material into cells, but it's usually as plasmids, and these are plasmids that will just sit in the cytoplasm, and they'll be expressed, but they won't stay there forever. They'll be degraded. Cells have a tendency to spit them out. So now in research labs, a lot of bacteriophages and retroviral vectors, there's a lot of interest in them because of the potential that you can use these vectors and you can use bacteriophages to introduce genetic material into the cell and then have this genetic material introduced into the host cell genome. So not just as a single circle of DNA that's floating in the cytoplasm, but actually have the genes stably integrated into the host cell genome. And this isn't hypothetical, this is actually done, and you may be aware of a therapy called CART, C-A-R-T therapy, or CAR T-cell therapy, chimeric antigenic receptor therapy, I think it stands for. I'm trying to remember now. The idea is that you can modify immune cells using lentiviral vectors. Okay, and you modify these immune cells, you introduce new genetic material to them, and by introducing this new genetic material, which is integrated into the host cell genome, you can force these immune cells to target cancer cells. So you can change these immune cells so that now, when they have these new genes integrated into the genome, they are able to target cancer cells. It's really quite amazing. It works. It's a confirmed therapy. It's been used to treat people with leukemia, I think. And that's using lentiviral vectors, but the principle will be the same for these bacteriophages. Okay, so those are the differences between the virulent and the temperate bacteriophages. Again, we're just laying the foundations here. We're just making sure we understand these life cycles. We're making sure we understand what these bacteriophages are. And what we want to do now is look specifically at the diseases these bacteriophages are implicated in. So again, we're at a point now where we understand that a bacteria, when infected by a bacteriophage, can potentially have genes introduced that are stably integrated into the genome, and these genes can potentially impact pathogenesis. So they can change the bacteria in such a way that it enables the bacteria to cause disease in a host, and we can track this down to specific genes. So what you're going to see now is we can have a bacteria and we can say this bacteria is non-pathogenic because it doesn't have this gene. Then we can have the same bacteria after a specific gene has been introduced into it via lysogeny, so by a bacteriophage, and then this bacteria has now become pathogenic. It is now capable of causing disease, and that's what we're going to discuss here. So just to reiterate here, we're saying that some bacteria become pathogenic when they gain genes through lysogeny. Also, importantly, equally as importantly really, some bacteria can gain antibiotic resistance genes by lysogeny. So we're talking about genes that can be introduced into the bacteria by bacteriophage. Obviously, it's a significant issue if pathogenic genes are introduced potentially, but also if we're having antibiotic resistance genes introduced, potentially bacteria are gaining the ability to resist antibiotics. They're gaining that resistance, and for obvious reasons, that's potentially a significant issue. Okay, So this diagram here, it's just an alternate diagram for lysis. It's not super complicated anyway, so I'm sure we understand how this lysis works. Okay, so lysogenic conversion, this is a technical term we use when bacteriophages are changed by the addition of genetic material through bacteriophage infection. So bacteriophage infects bacteria, bacteria acquires the genes, and we can then say this process is lysogenic conversion. We tend to use this term specifically when there's changes to the phenotype induced. So sometimes you could argue that bacteria will acquire genes that don't impact the phenotype of the bacteria, and that must happen, right? That's not necessarily lysogenic conversion. We tend to use lysogenic conversion to refer to the process that involves changing phenotype in the bacteria. So when the bacteria acquires genes that change the bacteria's phenotype in some way, and the examples we just looked at would be genes that enable the bacteria to cause disease in a way it couldn't previously, or that enable the bacteria to resist the action of certain antibiotics, so antimicrobial resistance genes. Crucially, this is heritable, so this is really important as well. So we've said that once a bacteria undergoes this process of lysogeny, or what we're defining it as here, lysogenic conversion specifically, because lysogenic conversion are the events that are really most interesting to us, right? Once a bacteria has undergone this process, it retains the genetic material over cycles of reproduction, right? So in the diagram earlier, we looked at how bacteria, once they have new genes integrated, they're able to reproduce, they're able to produce copies of themselves without losing that genetic material. Again, hopefully that was clear. And we therefore say the genetic material, these changes are heritable, they're inherited. And again, so the initial transfer is horizontal gene transfer, but then the genes are maintained vertically, so down successive generations of bacteria. Okay, so what we want to do now is look at some of the specific human diseases that are associated with this process of lysogenic conversion, so that are associated, that are caused by bacteria that acquire genes by bacteriophage infection. Here we have scarlatina, which is more commonly known as scarlet fever. It's exactly the same thing, just two alternate names. It's a bacterial infection, but as we're going to see, bacteriophages play a role. These are bacterial infections we're looking at, so don't make the mistake of referring to them as bacteriophage infections. They are bacterial infections. However, bacteriophages play a role in making the bacteria pathogenic. That's a really, really important distinction, because the bacteriophage isn't infecting the human host. That's not what's going on. It's the bacteria that is directly causing the disease. It's the bacteria that is infecting the human host. Okay, so back to scarlet fever. It's essentially a streptococcal infection. It's group A streptococcus. It can potentially develop from strep throat. That's how scarlatina tends to establish, and it's characterized by this red rash. That's how the name is derived, really. Scarlet. It's this red rash that can cover the face and neck, and then around the torso. Most common in children. You'll see the phrase streptococcal pharyngitis thrown around. It's caused by the same bacteria, as I kind of said. That's how it can start before it progresses. Pharyngitis, as we know, itis is inflammation. Pharyngitis is inflammation of the pharynx, which is caused in streptococcal pharyngitis by streptococcal infection. Strep throat is how we tend to refer to it. Again, it's just pharyngitis caused by streptococcal infection. So what's the pathogenesis of scarlatina? We've said bacteriophages have a role. So what's the actual role of bacteriophages? So to understand that, we need to understand how Streptococcus actually causes scarlatina. So that information is here. Like many other pathogenic bacteria, Streptococcus causes a special pathology when it releases a particular toxin, specifically an exotoxin. So we'll see a lot of different bacteria release specific toxins, and that's how they have their pathogenic effect. And this is something you may have come across in microbiology modules in the past. This idea that many bacteria release exotoxins into the local environment, and by releasing these exotoxins they cause disease because these exotoxins actively cause disease. And that's the case for Streptococcus in the context of scarlatina. So Streptococcus releases a protein called SPEA. This toxin is erythrogenic. It's encoded within the streptococcal genome. However, it is only encoded in the genome if Streptococcus has been lysogenized by a specific bacteriophage, which we call T12. So this is the principle that I introduced earlier playing out exactly. We have Streptococcus without the SPEA gene, then we don't have scarlet fever. It's not capable of causing scarlet fever. However, if we have Streptococcus that has been lysogenized, so it has been infected with T12 bacteriophage, then Streptococcus will be carrying the gene which encodes a specific toxin, and it therefore will be producing this toxin. It will be producing SPEA, and it will therefore be capable of causing scarlet fever. So when it's not lysogenized, it's not causing scarlet fever. When it is lysogenized with T12, it is potentially going to be causing scarlet fever because it has the gene for the toxin. Another example is cholera. So we've discussed, in past modules, you've probably discussed cholera. It's one of those really fundamental, well-known bacterial infections caused by Vibrio cholera. You may have heard the John Snow epidemiology study. So one of the first big epidemiology studies was conducted by John Snow on cholera. And the focus of that study is that cholera was being transmitted via the fecal oral route through contaminated water pumps. So people were drinking water contaminated with feces. As a result of this, they were becoming infected with Vibrio cholera, so they were developing the disease cholera, which is characterized by severe diarrhea, which is of course associated with fluid loss. So a big problem, still a big problem in the world today, not as much in Western countries, but certainly in countries that have their sanitation isn't at the same standard as Western countries. They perhaps don't have the infrastructure and things like that. It is spread by fecal oral, by the fecal oral route. So the bacteria are passed out, they're excreted in the feces, and then if they make it into a water supply or food supply, and then an individual ingests those bacteria, they can potentially become infected. So again, that is the fecal oral route. So it's understandable how things like sanitation and plumbing and sewage systems are what can potentially prevent the spread of cholera, and it's what's pretty much worked in the Western world. And this is what I'm referring to here. Again, I don't really want to go into detail. It's just a little bit of information. If you're interested in that original case study that was conducted by John Snow, it is interesting. So I put some resources here. Okay, and as I kind of touched on, we can see that currently cholera is largely confined to less economically developed countries, because these are the countries that don't necessarily have sewage treatment facilities. They don't necessarily have the plumbing and the sanitation that we have in Western countries. Okay, so what's the pathogenesis of Vibrio cholera? So here we are going to see the same principle play out again. We're going to see that Vibrio cholera gains the ability to cause a specific disease when it acquires a pathogenic gene from a bacteriophage. So this bacteriophage is CTX-phi, and CTX-phi is the Greek letter at the end, sorry. When CTX-phi infects Vibrio cholera, it can introduce the gene that encodes a specific toxin. That gene is CTX gene, and the toxin encoded is essentially just referred to as cholera toxin, which you may have heard of before in microbiology modules. Now, this cholera toxin is what directly causes diarrhea, so it's what directly impacts the enteric environment, it impacts the gut, and it causes the primary symptoms of Vibrio cholera. And therefore, it's clear to see how when Vibrio cholera becomes infected with CTX-phi, it's potentially gaining the ability to cause disease. The next example we have is diphtheria. Corynebacterium diphtheria is the causative agent of diphtheria, and you've probably heard of diphtheria before. Hopefully you're seeing now that we're talking about quite serious diseases, quite well-known diseases. So that's why we need to understand these bacteriophages. Even though it is a bit of a niche topic within virology, it's also super important we understand it because we're talking about really impactful diseases that it's really important that we understand the basis of. Okay, so diphtheria is characterized by like a thick lining at the back of the throat. It's like a grey coating. Some other symptoms that are associated with this, like bull neck. The significant problem is potentially that the airway could get blocked because we're talking about a really thick coating at the back of the throat directly caused by infection. Diphtheria is a really good example of a disease that has been well controlled by vaccination. So in Western countries, diphtheria rates have really dropped as a direct consequence of vaccination programs. So what's the pathogenesis of diphtheria? So like the other examples we've looked at today, Corynebacterium diphtheria can potentially be infected with bacteriophage, actually a range of different bacteriophages that all carry a specific gene, and this gene is TOX. This gene encodes a toxin that enables Corynebacterium diphtheria to cause disease. So it makes the bacterium pathogenic essentially. This diphtheria toxin, which is encoded by the TOX gene, essentially inhibits protein synthesis. So when this toxin is present, it's inhibiting cells in the host, so in the human host, from synthesizing protein, which obviously causes significant problems for the host, and this is associated with disease symptoms. Okay, next up we have dysentery, which is caused by Shigella dysentery. You may also see the pathology referred to as shigellosis or bacillary dysentery. We're talking about the same thing really. It's just because it's caused by Shigella dysentery. Shigellosis is a term that could be used. It's an infection of the lower GI tract. So as with a lot of these infections, we see severe diarrhea, and we see transmission from host to host via the fecal oral route very often. So what about the pathogenesis? Again, we're seeing the same principles play out over and over again. So just to reinforce why this matters, we're not just looking at one or two infectious diseases, we're actually looking at quite a few significant, important infectious diseases. So as with the previous examples, the causative agent, the bacteria, must be infected with a bacteriophage to acquire the ability to cause disease. And again, this acquired ability comes through specific genes that are required through the process of lysogeny. In this case, we're talking about Shiga toxin, which is STX, and it is acquired by Shigella dysentery when the bacteria is infected with a particular phage, which we refer to as the STX phage. And once this Shiga toxin is expressed, it inhibits protein synthesis, and in doing so, it causes the symptoms of dysentery, the principal one of which is severe diarrhea. Next up, we have botulism. Again, hugely important disease caused by Clostridium botulinum. We all probably know that Clostridium botulism, or we may know, sorry, that Clostridium botulinum releases a specific toxin, and that this toxin is what is responsible for the disease symptoms, many of which are types of paralysis. So you'll see the symptoms here listed. They're quite specific and quite serious, and they're all kind of associated with paralysis, with muscle paralysis, with localized paralysis. Infant botulism, this is botulism that occurs in infants, and the principal kind of feature of infant botulism is if you pick up a baby with infant botulism, they're kind of, it's described as floppy baby, like their limbs are just flopping down. So again, paralysis, that's the defining feature of botulism. So the pathogenesis, as we said, linked to paralysis. So what is actually causing this paralysis? It's specifically caused by botulism toxin, which is actually neurotoxin. The toxin can come in a couple of different forms, and it's encoded by different bacteriophages. But either way, Clostridium botulinum, to cause this disease, must have been infected with one of these bacteriophages, and it must have, through this infection, acquired the ability to express and create the botulinum toxin. As we said, it results in paralysis, and the reason it does this is because it blocks the release of acetylcholine, which I'm sure we know acetylcholine is a neurotransmitter. So if you're blocking the release of this, you're not getting stimulation of the muscles, which is paralysis, of course. So again, the real takeaway is that these bacteriophages are responsible for human disease, but not directly. Obviously, you can see these bacteriophages aren't causing the disease in the same way that viruses cause disease. However, they also are clearly implicated in disease. So that's why we really do need to understand these bacteriophages. When we're thinking about treatment for these infections, we're thinking about antibiotics. We're treating, again, it's the bacteria, because it's a bacterial infection. It's the bacteria that are directly causing the disease. So when we've been talking about viral infections, we've been talking about using antivirals. That's not the case in these infections. It's just not possible. It's just not going to work. Even if we had antivirals that treat bacteriophage infections, it's just not a reasonable thing for us to do, to go around treating bacteria for bacteriophage infections. It just makes much more sense to treat the bacterial infections, which is what we do. And that's why on most of these slides you've seen I've put up a little bit of information just indicating how we treat them, and it's usually antibiotics. There are issues with antibiotic treatment, however. So when these bacteria are lysogenized and they then encode the gene, or they then carry the gene that encodes a specific toxin, if you treat an individual with antibiotics, then you potentially kill all the bacteria. However, what you also potentially do by killing those bacteria is release huge amounts of toxin into the local system, and this is true from Clostridium botulinum, which we looked at a few slides ago. You have to think very, very carefully about treatment in cases of Clostridium botulinum, because if you're treating with antibiotics, you're potentially releasing huge amounts of toxin into the body system, which is problematic for obvious reasons. So again, the takeaway here is that we're treating these infections like bacterial infections, or clinicians are treating these infections like bacterial infections, not like viral infections. However, because these toxins are involved, there are some additional layers of complication to consider. Okay, finally, I want to just touch on something I mentioned near the beginning, this idea of using bacteriophages in research. Now, this is something we go into in depth, as I said, at the end of the module, but while bacteriophages are fresh in your mind, I want to just touch on the topic now and just make sure we have a general idea, excuse me, which we can then expand upon as we reach the end of the module. So again, as I said earlier on, the main principle here is using bacteriophages as a delivery method. So in research, it's very useful for us to be able to change the genetic material of cells. And I'll give you some examples in a second of why we might want to do that. We can do it using retroviral vectors because, again, they are capable of integrating genetic material into the host cell, but we can also potentially do it using bacteriophages because bacteriophages, like retroviruses, are capable of integrating genetic material into the host cell, and this is a concept we're going to explore again towards the end of the module. Now, this is beneficial for a number of reasons. One reason is conducting research in the lab. So we can think about therapeutics, and we are going to think about therapeutics. And I gave you the example earlier about using retroviral vectors to aid in the treatment of cancer. Very good example. Right now, I'll give you some research examples. So you can be in the lab and you can want to change the genes that a cell expresses. So why might you want to do this? Well, I can say because it's something I've done myself a lot. It's something my students have done. As you probably know, I work on HIV and I specifically look at proteins that inhibit HIV infection. So one of the things that is useful for me to be able to do is I can force a cell to express large amounts of specific protein. Okay. If I can force a cell to express large amounts of specific protein, I can then challenge that cell with HIV. So put HIV onto that cell and then do the same for a cell that isn't expressing that protein, which is my control, and then I can measure HIV infection. If the protein that I'm forcing the cells to express is fighting HIV, so if it inhibits infection, we should see infection go way, 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 way down. And I have multitudes of graphs looking at this where I'm forcing these cells to express the protein and I'm not forcing these cells to express the protein. And then I infect both cells with HIV. So this group of cells and this group of cells, then I measure infection. And in these normal cells, infection comes up to whatever, that's my control. And then in this treatment group, I'm looking at whether infection is higher or whether it's lower. And that's how we would determine whether this protein has had any impact on infection. That's just one example of why I have an interest in doing these techniques, or techniques like these. I actually use lentiviral vectors, but again, it's this principle of changing the genetic material of cells, and there's a whole multitude of other reasons individuals would want to do this in the lab. If you just want to purify a protein, so if you have a protein that you don't know the structure of, which is something else I've done, but some people will base their entire careers on this. They will be figuring out the structures of proteins. It's really useful, important work. They can force cells to express large amounts of this protein, and then they can isolate this protein, and then they can purify it, and then they can use these huge amount of protein that they've isolated and purified to experiment on so they can determine the structure. So again, really, really useful to be able to force cells to express the genes we want them to. And we've had a look at a few examples there. We're going to look at additional examples towards the end of the module, and crucially, we're also going to look at how these bacteriophages can be used therapeutically. So there's two real ways in which we can use bacteriophages for our benefit. One, research, as we've kind of just discussed, to medicine. Okay, so I gave you the cancer example, which is a valid example, but there's even clearer examples of how bacteriophages can help medically. And it's kind of intuitive if we think about it. So we know these bacteriophages, even the lytic ones, are capable of helping us therapeutically. And the reason for that is these bacteriophages infect bacteria, as we've talked about. Some also infect archaea, that's fine, but we're really concerned with bacteria. So if we think about the lytic bacteriophages, these infect bacteria and these lyse the bacteria, okay? Straight away we can hopefully see the therapeutic benefit of that. Bacteriophages infect bacteria, they don't infect eukaryotic cells. That means if you have a bacterial infection, you can potentially treat that bacterial infection with bacteriophages, because those bacteriophages are going to lyse the bacterial cells without doing anything to the host cells. And currently there are a range of therapies being developed that utilize bacteriophages in this way. So they use bacteriophages as a treatment or as a preventative measure for bacteriophages, for bacterial infections. And the principle is what we've just described. The principle is the bacteriophages, and we're talking about lytic bacteriophages now. They will infect bacterial cells and they will lyse those cells, but they will not harm host cells. And we're going to look at some examples of the development of these. Again, what they're called is phage therapies, and we're going to look at some examples of those towards the end of the module. Okay, thanks very much. I hope you found the lecture interesting and useful, and I'll see everyone Monday morning. Thank you.